Hello everyone, my name is Julie Fan and I work in the Harris lab with Dr. Samantha Harris. Our lab's overall goal is to better understand a protein called a cMyBP-C, and what this protein basically does is it regulates heart contractions, and I'll go into this more in the introduction. But another thing you should probably know is that another name for cMyBP-C is C protein, so I'll use both terms interchangeably to refer to the same protein. And so to get right into it, the title of my presentation is Reduced Expression of cMyBP-C in Left Atria Relative to Left Ventricle of Cat and Mouse Hearts. So we took tissue from cat and mouse hearts and compared how much C protein was found in the left atria to how much C protein was found in the left ventricle. And the atria and ventricles are just two different structures or chambers of the heart, and these chambers have different structural and functional variations, and we think C protein plays a role in these differences. So that's what we sought out to explore. The first thing you should know is what C protein is. So cMyBP-C stands for cardiac myosin binding protein C, and just looking at what the name sounds like basically tells you what it is. So cardiac tells you that this protein is found in the heart, and myosin binding tells you that this protein is found near another protein called myosin, which many of you may know is an essential component of muscle fiber. It basically allows for heart contractions. So cardiac myosin binding protein C basically tells you it's a protein that regulates heart contractions. And so specifically, it regulates the strength and the speed of heart contractions, and mutations in the gene that encodes for this protein are linked to many disorders of the heart, but most prominently hypertrophic cardiomyopathy, or HCM, and heart failure. And this just briefly shows you an image of where C protein is. So this is a muscle sarcomere representational version, and in orange are chains of C protein. And you can see one end of C protein is embedded in this blue thick filament, which is myosin. And then there's also a free-floating end, and this free-floating end interacts with other nearby proteins to exert its regulatory role, C protein's regulatory role. For instance, it interacts with actin, titin, even myosin itself, and a bunch of other proteins. And so now you have a basic understanding of what C protein is. What is the purpose of this presentation? So, like I said, our lab's goal is to better understand C protein, and in this project, what we wanted to explore is whether the amount of C protein that's expressed is different or the same in the sarcomeres of the atrial chamber compared to the ventricular chamber. And so we specifically hypothesized that C protein expresses differently in the atria compared to the ventricles. And there are multiple rationale for this. For example, our two main ones are, first of all, as we mentioned, C protein is a regulator of heart contractions, and different heart chambers have different contractile properties. For example, ventricles eject far more blood and contract far with greater intensity than the atria. And so because they have different contractile properties, they might have to be regulated differently, meaning C protein might have to be expressed differently in some way. And in our hypothesis, that difference is the amount that is expressed is different. So that's one reason. And then another reason we think C protein is expressed differently between the atria and ventricle are that many mutations in C protein's gene, as we mentioned, are linked to HCM, hypertrophic cardiomyopathy, which is hypertrophy or excessive growth in the walls of the left ventricle, which is interesting because this protein is found in all the heart chambers, yet somehow it's linked, many mutations in this gene are linked to a disorder that only affects the left ventricle. So that again leads us to believe that perhaps there's some difference, and that difference might be a difference in the amount expressed of C protein between these heart chambers. So that's our main hypothesis, that C protein is expressed differently in the atria compared to the ventricles. We had a side hypothesis that has a little bit to do with the second rationale, which is we wanted to test if a specific mutation linked to HCM, called the A31P mutation, alters relative chamber expression of C protein. And what this basically means is we wanted to look at mutated cats and how C protein is expressed in their heart chambers. And compare it to non-mutated cats to see if there's a difference. And so to test this, both of our questions, we'll go into the methods. And the main objective for all of our tests is to measure how much C protein is in the left atria, how much is in the left ventricle, and make comparisons, 2 different types of comparisons for 2 different tests. And in the essence of this project, we relied on Western blots, and we took tissue from mice as well as 2 types of cats. We used all of these heart tissue replicates, biological replicates, as well as technical replicates to answer our main question, and then we looked deeper and only compared the cats for our side question, with our wild type being the non-mutated cats and the homozygous being the cats with the A31P mutation. And to go deeper into the methods, first we had to prepare our tissue samples. We made homogenates, which was the whole heart tissue, so it included all types of proteins. And then we did SDS page Kumasi gels to standardize the loading before we moved on to Western blots. And then with our Western blots, we did quantitative Western blots by normalizing our C protein, which is in green, to actin, which is in red. And what Western blots basically do is they measure how much protein is in a tissue sample, how much of a specific protein, in our case, C protein, is in a tissue sample, because we selectively had antibodies that looked at C protein, which were the green antibodies, and then we used the actin just as for normalizing. And then, so we ran 3 technical replicates, 3 blots for each animal, which are biological replicates. And we organized all the data together to answer our 2 questions that we mentioned above. I don't know if I can zoom in. Apparently, yes. Okay. So then moving on to our results to answer our 2 questions. So the first question was, does C protein express differently in the left atria compared to the left ventricle? And so our null hypothesis would be that no, there's no difference between how much C protein is expressed in these 2 chambers, in which case the value would be 1. After we pulled all of our data, we divided each LA left atria by its subsequent LV or left ventricle. And if the same thing is divided by itself, it would be 1. So that would be the expected mean if C protein expressed in unequal amounts. And then, however, as you can see in this graph, in all of our different populations, sample populations, in the mice and our both types of cats, they're significantly below that 1 threshold. And we did a 1 sample t-test to see if it was statistical, and we determined based on those results that yes, they were statistically significant, and we were able to conclude that yes, C protein is expressed significantly less in the atria compared to the ventricles. And then our second question was about the mutated cats and whether those cats expressed C protein differently relative across their chambers compared to wild-type cats. And so in this study, we only looked at the cats, so we compared them to each other instead of to an expected mean. And in this case, we did 2 sample t-tests, and based on our results, there was no significant difference between the wild-type cats and the homozygous cats. So to move on to our conclusion, the left atria showed significantly reduced C protein compared to the left ventricle based on the first graph that I showed, and our 1 sample t-test, which showed significant difference. And so future directions would be to determine what that reduced expression equates to functionally in the heart, and if it contributes to the functional differences and the pathological differences between the left atria and the left ventricle. And then we can also look at our second finding, which is that the homozygous cats with the A31P mutation did not affect relative C protein expression compared to the non-mutated cats, which suggests that the A31P mutation does not alter relative C protein expression. And this is just one of many HCM mutations, so moving forward we can test other mutations, see if there's a link, or we can widen the scope and explore how C protein is expressed and what the amount is in regards to other cardiomyopathies, such as atrial fibrillation. And that can help us better understand C protein's role, as well as C protein's role in the heart, but also how these disorders develop over time. And that can help us for prevention and cure development. Furthermore, just in general, we can explore more than just in future directions, we can explore more than just the amount of C protein that's expressed. It's a good starting point to start to figure out structurally how C protein differs between the chambers, but we can also look at the location as another aspect of structural differences in C protein expression between these heart chambers. We can also look at maybe different phenotypes, and there are a lot of different possibilities, but that's where we're at in the discussion of this project. So that's the end of my presentation. Thank you for listening. If you have any questions, I think SARCEF, the website, has areas where you can comment. And I also want to have a quick acknowledgement for the members of the Harris Lab, especially Dr. Harris, Rachel, and Saul for helping me through the project. And this was also in partnership with UBURP, so I'd like to thank the UBURP program as well. Thank you.